Um, Hello, my name is John Mark, and this is my group's SRC presentation. This is my group's SRC presentation on the investigations of biological nitrogen fixation in the Zoomatic Land Act. So I have a partner, but she kind of needs there, can't go by myself today. All right.
Well, we do have these here. You want to get out of the
So biological acidification, so the process, in which N2 turns into ammonia. This process is fixed by an autogenous enzyme within a bacteria known as azotrophs. For our model bacterial system, for our experiments, we use the bacteria landiac. So this process turning N2 into ammonia, this measuring this atmosphere turning into ammonia, is crucial in the outgoing process. So as you can see, this is the reaction that takes place. It requires 16 ATP and 8 electrons to only create two ammonia molecules. So this process is pretty inefficient. The autogenous enzyme. So as you can see, the enzyme is made up of OP protein, which is the green and pink that you can see in the middle, and also made up of iron proteins, which is the two blue proteins to the side. It also contains three metalloclusters, which are a cluster of metals that sit in pockets of the enzyme. The first one you see the hemo cofactor, which sits at the active site of the enzyme where the N2 binds to convert it to ammonia. And the 4 iron 4 sulfur polycluster that shoots electrons from the iron protein from the blue into the P cluster in the MOP protein using ATP, which is crucial for the reaction to take place. So the way we see this bacteria is through a process of cryo-electron tomography, which also called cryo-ET. There's basically an electron microscope that creates images from different tilts and angles of the object to a 3D image, and so we can see structure in the cells. The cryo-ET samples must be 300 nanometers thinner. The electrons to be scattered and detected, but the bacteria landiae are around 2 microns thick. So it's too thick to see under cryo-ET. So we use a process called focused ion beam milling. It's basically like shaving off slices of the cell to make it thin enough so you can see under a microscope. So we noticed two major things using the cryo-ET. We saw the cell contained a lot of vesicles, which is pretty unusual for all bacteria. So we saw the vesicles seem to be created by peeling from the inner membrane. And we saw the vesicles only appeared in bacteria with expressing nitrogen. We also saw an abundance of filaments in the cell. So there seems to be an odd relationship between the nitrogenase and the vesicle itself. So that's what we're looking at. So as you know, the vesicles are abundant in bacteria, but their purpose is abundant. Which leads us to our motivation for our next experiment, which was to isolate the vesicles from cells expressing nitrogenase and determine their protein contents. Determining their protein contents would reveal the vesicle's purpose. So we have two ways of isolating these vesicles, and one being osmotic lysis. So what we do here is we place bacteria in a glycerol solution until it becomes fully homogeneous. We spin down the solution using a centrifuge, which creates solid pellets of bacteria. Then, for the solid pellets, we put that into a lysis buffer, which is similar to the vegetable solution, but without the glycerol. So the imbalance of the glycerol between the inside and outside of the cell causes water to rush into the cell due to osmosis, which causes the cell to burst. We also add DNAs, and then that breaks down the remaining DNA floating in cell cytoplasm, and that's fine. And then that breaks down how to digest. So basically what we're doing is we're eating away at anything else but the vesicles, so we can see it as best as possible in our images. We also use a process of chemical lysis, involves using a emulsiflex, which you can see in that picture on the right. That highly pressurizes the atmosphere the cells are in to force them into fitting through an incredibly narrow space, essentially forcing the cells away. So from the image we see here, I just want to make note that we didn't see a lot of vesicles in our images, but you can see that there are very clearly intracellular vesicles. But in the other image with the mechanically lysed, this is automatic lysed images. But when we use mechanically lysed prep, we noticed that these weren't clearly intracellular vesicles, but it's because we saw pieces of the edge layer of bacteria, which is really on the outer membrane in our images. So these vesicles weren't clearly intracellular; they could have been just ripped apart and floating around, not necessarily in the cell. So for our conclusion, we determined that we can isolate vesicles with single membranes from automatically lysed other bacterial and cells. However, like I said, they are low in abundance. So the reason they are low in abundance is because that most of these vesicles are still attached to the inner membrane when they peel off. So for experiment 2, we wanted to isolate these vesicles still attached to the inner membrane from cells expressed in nitrogenase and test for interaction with nitrogen. So the way we did this is we followed a shoe and meat 2023 protocol, in which we used a sucrose gradient method to separate the inner and outer membrane of bacteria based on their respective vesicles. We took one bacteria that wasn't expressing nitrogenase, since we fed the cell nitrogen, the one that was, the one we kept from nitrogen, and placed these both into sucrose gradients. We then put these two into an ultracentrifuge, which allowed for the sucrose gradient to separate the inner and outer membrane based on their respective different vesicles. We then took the inner membranes of the expressing and non-expressing bacteria and ran those through a gel, also known as gel electrophoresis, in order to compare the different proteins in the individual bacteria. In the gel, the smaller proteins pass through the gel matrix easier and fall down the gel, while the bigger proteins stay close to the top. The ladder that we see to the left are standard molecular protein weights we can use to compare our models. As you can see, there are similar proteins found in both, and they're pretty identical. However, the electrophoresis is not a very sensitive method. Therefore, we have also prepared these samples for a mass spec, and we see that there were 97 proteins unique to the inner membrane, although they're mostly transporter proteins. Plus, there are also a high number of uncharacterized proteins from body as well. So nitrogenase, as I've spoken about, converts nitrogen into ammonia, but it also does it converts acetylene into ethylene, which is pretty easy to detect by Gaussian photography. So what we did was we measured the ethylene production of various things. The inner membrane alone, which of course would produce nothing. The inner membrane and the MOFE protein being a part of nitrogenase. The inner membrane and the Fe protein. And in the inner membrane and nitrogenase and just nitrogenase. And as you can see, just nitrogenase and the inner membrane plus nitrogenase produce the same amount of thing. So we came to the conclusion that the inner membrane didn't affect the production much, meaning there wasn't much relationship between the nitrogenase and the inner membrane. So we concluded that we are able to isolate the inner and outer membrane fractions using sucrose gradients. Mixing inner membrane fractions with the nitrogenase proteins does not affect their activity. So for experiment 3, we want to examine if there's a relationship between the type 6 secretion of the sheath protein isolated from the Lactobacter landi and the nitrogenase proteins. So as before, filaments are abundant in the Lactobacter landi bacteria, and these are known as type 6 series of filaments. So what are these filaments? These are filaments that harpoon out of the cell and secrete bacterial toxins into other cells. This is odd for a zoobacter to have, since it doesn't have much competition and it is a pathogenic bacteria. After flinging into other cells, the filament leaves behind an outer sheath, almost like a bow, if you will. I had a video up. Oh, shoot. I had a video up today, so I'm not sure it's gonna work. This is how the building kind of works. That kind of comes out of the cell and at least behind that kind of sheet. That's what we focus on. Yeah. Yeah. Pretty cool. So. So we want to check for association between the outer sheet that's left in the bacteria and the nitrogens. So we took isolated filament sheets and mixed it in solution with a MOFI protein. MOFI protein is just a part of nitrogenase without the iron protein. We see the filament to the annulation with nitrogen fixation. We put the solution to a column containing resin, having smaller molecules releasing large ones to the column, kind of the opposite of the electrophoresis. If these substances interacted, we see one large streak because they form one solution. But we see instead the filaments in the MOFI protein all go through the column independently because we have multiple peaks on them. So far, we concluded that C6 acetylcysteine protein filaments do not form a stable complex with nitrogenase MOFI protein that can be isolated by scientific chromatography. However, it should perform with the nitrogenase FE protein, just the other part of the nitrogen. So, our overall conclusion was that from our preliminary experiments, it did not appear that the vegetables and the filaments have direct interactions with the edge proteins, but there may be indirect feedback between the systems within the bacteria and like that. So, thank you so much for listening. I'd like to acknowledge Rees Lab, Clemens Lab, and yeah, thank you so much for listening. Have any questions? Yeah. So it's an enzyme, so it basically just like catalyzes the reaction. So the actually will pop. What? I'm not entirely sure like how the enzyme kind of how the chemistry works exactly, but all I know is that it catalyzes the reaction and it allows for energy to.
It's actually a great question because we don't really know how this enzyme works, but we generally know that electrons are transferred from the iron protein to the MOFI protein, and then that nitrogen is reduced at that chemo cofactor that I was pointing out. But there's probably some transformations that take place in the active site, and we're really not sure.
Thank you. Great question. Any other questions?
Just wanted to talk a little bit about what surprised you or what was your favorite part of the experience?
So this is my first time working in a lab. And it's obviously something, it's something that I want to pursue in the future, biology. And I think this was a really big opportunity for me to like learn what it's like to be in a lab, having things like that, knowing like different definitions of different things. It's going to get my way around it. So this is like, that was one of my favorite parts, staying in the lab, learning how to do basic things, you know, building my knowledge for the future. You know, so. Thank you.